Good afternoon and welcome to you all for this Adelaide Biomed City Minireview webinar series. My name is Andrew Zannettino and I'll be your chair this afternoon. Before I introduce our speakers for this afternoon, I'd like to acknowledge the Kaurna people, the original custodians on the land on which we meet here today. I'd like to pay my respects to elders past, present and emerging. And before we kick off, I'd just like to remind people that this is an interactive opportunity and you are able to ask questions of our speakers. I'd ask you to use the Q&A function on the bottom of the banner, or alternatively, you can use the chat function. And that'll allow me to relate the questions to the speakers. This afternoon, we'll hear from two speakers from the University of Adelaide whose research focuses on cancer in women. And our first speaker this afternoon is Professor Martin Oehler. Professor Martin Oehler is the Director of Gynecological Oncology, I should say, at the Royal Adelaide Hospital. Martin is also the Clinical Professor in the discipline of obstetrics and gynecology at the University of Adelaide. As you'll hear this afternoon, Martin is a clinician and scientist with a long track record of contributions to both basic and translational ovarian cancer research, which are his main areas of research interest. Today, Martin's presentation title is Development of Novel Approaches for Ovarian Cancer Diagnosis and Treatment. And I'd like you to welcome Martin to the screen. So thank you, Martin.
Yeah, thank you for the kind introduction and for giving me this opportunity to talk about our ovarian cancer research. About 1,700 women each year are diagnosed with ovarian cancer, and about 1,000 women die from the disease in this country. So it's a significant health issue which causes serious grief to the community. So it's a cancer with very bad prognosis and high mortality, and about every 8 hours one woman dies from ovarian cancer in Australia. So it's a serious problem. Well, the two main reasons why ovarian cancer has such a bad prognosis is that ovarian cancer is usually diagnosed at a very late stage. There is no early detection test. So, and when the cancer is very advanced, it's very difficult to cure. So there's really an unmet need for an early detection test. And the other reason why ovarian cancer has such a bad prognosis is because despite radical treatment consisting of surgery and chemotherapy, most patients at some point develop a recurrence, and when the cancer becomes chemo-resistant, then really leads to a fatal outcome. So the main challenge of ovarian cancer is early detection and novel targeted treatments to overcome chemoresistance. So in about 2006, Peter Hoffman from the Clinical Health Sciences at UniSA and Camilla Riccardelli from the discipline of Obs and Gynae at Adelaide Uni, we got together and decided to do research into ovarian cancer, really to improve the outlook for women with this disease. And I'm confronted with ovarian cancer every day because I operate on these patients. So over the years, more and more research groups joined us, and we are now quite a diverse, multidisciplinary group of researchers, all working towards the same aim, really to beat ovarian cancer. So it's a mix of researchers, molecular biologists, immunologists, also clinicians, and so we are quite a productive group which tries to progress research into ovarian cancer. In 2019, the group then initiated a research initiative with the name Development of Novel Approaches for Ovarian Cancer Diagnosis and Treatment. And this also included a major ethics application because a lot of our research is really based on clinical samples. And it included several areas like early detection, chemoresistant, therapeutic targets, personalized treatment, and therapeutics. So I will now very briefly talk about some of our research projects, but obviously it will be very brief. So we are very happy to be contacted if you want to have more detailed information. So one of our main areas of research is really the development of an early detection test. And one of our big projects was to develop an early detection test based on autoantibodies. So we used an immunoproteomics approach to detect autoantibodies which are upregulated in early ovarian cancer. You want to detect something which tells you a patient is suffering from early disease, because that's the stage where you can treat and cure a patient. We identified several candidates which are very sensitive and specific for ovarian cancer for early stage disease. And when we validated, then really we found that they have the potential to be used for development of early detection tests and subsequently for screening tests. So what we're currently doing, we try to optimize an assay. It's a so-called Luminex assay, and we also do some epitope mapping to make the test more specific. So once this assay has been established, we'll perform a prospective study, and then hopefully it's going to lead to population screening. Peter Hoffman and myself, we founded a startup called OncoDX because you need very good funding, and this we're going to try to achieve now by some investors to drive this research forward. So it's quite promising, exciting research. Another project we did to identify other interesting markers or potential markers for early detection was we took bloods, or I took bloods intraoperatively from ovaries, malignant ovaries, and we were able to identify new markers which are higher concentrated in the blood directly taken from the cancers, and they're also now under investigation. Another interesting protein we looked into is annexin A2. We measured this marker, this protein in the bloodstream of patients with early ovarian cancer, and could found that this marker might also be suitable for early detection. Annexin A2 is a very interesting protein, which our group had discovered several years ago when looking into new targets for ovarian cancer metastasis. We could actually show that cancer which grow in mice growth can be inhibited by blocking annexin A2 with an antibody. So that was quite interesting to see that projects in early detection can also lead to progresses in the therapeutic area. Another pathway which was targeted in the past, and this project was driven by Frank Grützner from the Genetics Institute at Adelaide University, was a project into the PV interacting RNA. And more recent research initiative, which is done by Melissa Pittman from Molecular Life Science at Adelaide Uni, by Stuart Pitson from the Center of Cancer Biology, is looking into a sphingolipid pathway, which is also a very promising area to develop therapeutics. To get a good animal model, Melissa is currently developed a patient-derived xenograph model, which is going to be very useful to test compounds or new therapeutics in that area. An area of intense research in our group is also chemoresistance, and our group has explored the role of the extracellular matrix protein hyaluronan on chemoresistant ovarian cancer. That's been done for many years, and Carmela Riccardelli is an expert in these topics. Latest results have now shown that hyaluronan has certain molecular weight proteins which affect chemoresistance, and these certain molecular weights, they upregulate factors such as DAP2, and DAP2 is known to affect prognosis in patients. So it's quite an interesting area, and this research is currently written up for publication. Another project which looks into chemoresistance was based on Peter Hoffman's MALDI imaging technology. So what we did, we compared tissue from patients who responded to chemotherapy versus patients who don't respond. And then we did a differentiated proteomics analysis, and we were able to identify markers which are specifically upregulated in chemoresistant disease, and they might potentially be targeted to reverse the process of resistance. Another project which out of the area chemoresistance, but more into looking into personalized treatment, is a project which aims to develop a chemosensitivity assay, which predicts who might respond to treatment, who won't, because obviously giving the chemotherapy is not effective causes morbidity, and obviously also potentially affects prognosis negatively. This project is driven by Manuela Kingler-Hoffman from the Clinical Health Science at UniSA. And what she uses spheroids from ascites, and these spheroids are then analyzed by MALDI imaging technology. So it's a quite exciting project which is under development and is quite this potential impact on patient management. The last area I'm going to talk about is obviously the therapeutics area. And the major effort goes into the development of a novel CAR T-cell immunotherapy against ovarian cancer. This project is done together with Carina Biotech and also Simon Berry from the molecular immunology at Adelaide University. So we've been quite successful in developing a CAR T-cell immunotherapy against a receptor called P2X7, which is specifically mutated on cancer cells. This was shown to work in vitro and in vivo. And another target we are now focusing on is the CAR T-cell therapy. It's a receptor called LGR5, and it's also shown in vitro as quite an effective target. The last project I briefly want to talk about is research conducted by Shudong Wang from Pharmacy and Medical Science, UNISA. She successfully developed a novel group of therapeutics called CDK4 inhibitors, and these inhibitors are now tested already in phase 1 trials, so that's quite exciting. So she's currently running phase trials in phase 1 trials at Flinders Medical Center. So this was briefly what we're doing. So what are the key capabilities of our research group? We're obviously a multidisciplinary team. We have experience in a wide range of skills, basic clinical and translational cancer research. So we have the appropriate mix to be quite successful in ovarian cancer research. We have also access to very high quality gynecological cancer samples because I collect them during surgery. That's also a real plus which we have. We also have a tissue bank, all obviously approved by ethics. So that's the strength of our unit. And we have well-established ovarian cancer models which we have used for many, many years. And finally, we have access to a first-class state-of-the-art proteomics facility with Peter Hoffman. So we're always happy to collaborate. We're always quite excited to get new skills into our mix. So if you want to join us, please send us an email, and we'll get in contact with you. So thank you for your attention.
Thanks so much, Martin. That was a fantastic presentation and really an example of a really true example of multi-institutional forces coming together to address a really critical key cancer in women. So Martin, if I might start off with the first question, if I could. I'm really fascinated by the auto-antibody test that's in development with your startup company, OncoDX. Just interested to know whether auto-antibodies are a feature of other cancers outside of ovarian cancer, to your knowledge, and in fact, do they provide a very good early indicator of the cancer initiation process within the ovary?
Yeah, that's quite fascinating actually. Autoantibodies have also been described in breast cancer. The interesting thing about ovarian cancer is actually that have been reported to start raising in ovarian cancer before actually a malignant lesion is identified. So it's already, the immune system somehow is already being able to detect when the precancerous changes occur. So some kind early in oncogenesis. So and that makes it such a how to say, promising way of early detection, because it lightens up before actually the patient has a macroscopic lesion. And the problem with ovarian cancer is you really have to detect the disease extremely early before it can metastasize, which is very early in the disease process.
So in terms of metastasis or metastatic process, where are the secondary organs that are affected by ovarian cancer?
So in ovarian cancer, the cancer cells shed off the ovary or the fallopian tube and then circulate through the abdominal cavity. Right. And then they implant on the peritoneal surfaces, and the patients then develop ascites. Right. And then get very sick. And then it's usually disease is confined to the abdominal cavity, but the cancer starts coating the bowel, which then ultimately leads to a bowel obstruction, which is a terminal event. And the difficulty is to, once the cancer spreads to the abdominal cavity, to remove it surgically because it coats vital organs. And that's the reason why we always combine the treatment with chemotherapy and surgery, or vice versa, because we know that chemotherapy is very good in eliminating a certain percentage of the cells.
Has any studies been performed to look at the organoids that are developed from the ascites versus the primary tumor? Are there any clear differences between
yeah, so
those
that's
tumor
actually
types?
been shown. Actually, it's fairly recent research over the last few years that actually these spheroids. So the cells don't metastasize as single cells, but they form these spheroids, and these spheroids actually have certain properties which enable them to survive metastasis and to implant and to promote the invasion process. So that's been fairly recent research, but it's showing a huge difference. Also a niche of resistant cells. So these organoids are an area of intense research and a promising area too to make progress.
Fantastic. Look, we have a question from one of our audience. It's Munir Ahmed. Martin, it was a wonderful talk. Have you done any PET imaging studies to look into some of these markers you talked about? We have expertise in new PET tracer development for specific markers for CNS and oncological disorders at SAMRI.
No, so we haven't done any imaging studies, so would be very happy to look into that area and to collaborate. Yeah, no, thanks a lot.
Wonderful. Thank you for the question. Look, that brings us to the end of the first speaker. Thank you, Martin, for your presentation. It was a really fantastic talk, and allows me to introduce our second speaker for this session this afternoon. Our second speaker is Associate Professor Theresa Hickey. Associate Professor Theresa Hickey received her PhD in medicine at the University of Adelaide in 2006. Theresa is currently a National Breast Cancer Foundation Fellow and heads the Breast Cancer Research Group at the Dame Roma Mitchell Cancer Research Laboratories, located on the 8th level of the Adelaide Health and Medical Sciences building. In addition to her wonderful research, Theresa has been acknowledged by being awarded the 2020 Executive Dean Awards for Mid-Career Research Excellence, and for her work published in Nature Medicine in 2001, which has resolved a major clinical controversy about targeting androgen receptors in estrogen receptor-driven breast cancers. Importantly, this research has actually informed several current clinical trials. Today, Theresa's presentation is entitled Patient-derived Models and Multi-omic Technologies to Investigate Sex Hormone Receptor Functions. Welcome, Theresa.
Okay, thank you, and good afternoon, everyone. I'll just start by reminding people that sex hormone receptors are ligand-activated transcription factors that bind to DNA on hormone response elements to regulate target genes. These hormone receptors are all very related, and the three key ones are the estrogen receptor, progesterone receptor, and the androgen receptor. Now, the estrogen receptor alpha drives up to 80% of all breast cancers. And of these breast cancers, about 60% are positive for the progesterone receptor, and almost all, up to 98% of ER-positive breast cancers are positive for the androgen receptor. And our laboratory has been very interested in looking at the interaction of these three signaling pathways. Importantly, ER-positive breast cancers are treated with a variety of different anti-estrogenic drugs, but therapy-resistant disease is still a major cause of death from breast cancer, and anti-estrogenic drugs really do make patients feel miserable, and so alternative therapies are really necessary. And we've been working on a completely different alternative therapeutic strategy in which we don't inhibit estrogen receptor in the classic way that anti-estrogenic drugs do, but we try to reprogram it via activation of these parallel sex hormone receptor pathways. And we believe that this strategy is going to be less prone to development of therapy resistance because these are endogenous mechanisms that regulate breast homeostasis, and also that they will be associated with less therapeutic side effects. Now, our first foray into this line of investigation was this paper published in Nature in 2015, showing that the progesterone receptor can modulate ER alpha activity in breast cancer to reduce cancer growth. This really quickly led to 3 clinical trials within 2 years of publication, in part because there are many PR modulating drugs available for clinical repurposing. And importantly, one of the other aspects other than inhibiting tumor growth is that it's been shown that this strategy counteracts hot flushes associated with anti-estrogenic drugs. Now, I'm not going to talk much about that study for time, but I'm going to move into what Andrew mentioned was our most recent paper published last year, where we showed that the androgen receptor is a tumor suppressor in estrogen receptor positive breast cancer. And as he said, it resolved a major controversy about targeting the androgen receptor, as many people thought because the androgen receptor drives prostate cancer, that it must be a bad factor in breast cancer as well, and that we should use androgen receptor inhibitors. But we showed that you needed to stimulate the androgen receptor, and we used a selective AR modulator called enobosarm that is now in 2 clinical trials for advanced metastatic ER-positive disease. Importantly, this drug enobosarm lacks virilizing activity in women and has quite beneficial effects on bone, muscle, and mental health. And I'm summarizing a very large compendium of mechanistic studies in this schematic just to illustrate the general concept is that when the estrogen receptor is on its own and driving breast cancer growth, it binds to DNA associated with key oncogenes that drive breast cancer growth. But when we stimulate the androgen receptor with an agonist, that activated androgen receptor is able to displace the estrogen receptor from these DNA binding sites. And in part, what we see is that it takes the estrogen receptor along with it to certain target genes, but also one of the ways androgen receptor action by sequestering these key co-activators to upregulate another set of AR target genes. And collectively, this new transcriptome that gets activated in the presence of the androgen receptor suppresses growth of the ER-positive breast cancer. Now, I'm going to just briefly give you snapshots of some of the patient-derived models we've used to support this work and the ones that we're developing. One of the first is a patient-derived explant model in which we take either normal or cancerous breast tissues from the patient and we grow them on a gelatin sponge support to which you can add drugs to the growth medium. And then you can do downstream analyses of these tissues. For example, here is showing you uptake of BRDU as a marker of proliferation, or here showing you harvest of the tumors to do RNA-seq and transcriptional profiling, collectively to show here in terms of proliferation marker, or here in terms of hallmark cell cycle genes, that estrogen alone will upregulate proliferation, but when you add the androgen, in this case dihydrotestosterone, that opposes that estrogen action in terms of proliferation and downregulation of ER target genes. Now, another model that we've used in these studies are patient-derived xenograph models of breast cancer. These are generally developed from metastatic sites in patients, and the mice and serially passaged in mice, and the mice become avatars. Importantly, most of these models are propagated in the memory fat pad, and I'll explain in a minute why that's important. But this is just 2 growth curves that we generated as part of our study with 2 different models of ER-positive breast cancer, very advanced disease that has ER mutations that drive therapy resistance, showing you that estrogen stimulates in vivo growth of this model, but when you stimulate the androgen receptor with either dihydrotestosterone in blue or enobosarm in purple, you see quite potent and prolonged growth inhibition. Now, we use a variety of techniques to mechanistically interrogate the tumor samples from these experiments. We've used ChIP-seq for cistrome profiling and RNA-seq for transcriptome profiling. ChIP-seq for looking at under different treatment strategies, what happens to the binding of AR or ER on DNA. And these are just some examples. And then, of course, you can look at the transcriptional consequences of that by RNA-seq. We also have used this technique called Q-plex rhyme to do protein interactome profiling. And this is where ER interacting proteins are identified in breast tumors or breast PDX models to try to identify and interrogate what are the key interacting proteins that are influencing ER activity in a tumor or any given model. Now, one of the things that we're working on now is to develop a much better model of that better models breast cancer progression. And this is a technique called mammary interductal xenografting. So instead of establishing the tumor in the mammary fat pad, in this technique, we take the tumor cells, we label them with a fluorescent or a bioluminescent dye and inject them right into the mammary ducts of the mouse. So you establish the tumors where the breast cancers would naturally develop, and you can observe disease progression from in situ to invasive, and importantly to metastatic disease stages. And this is just showing you a picture of one of the models we have up now. We have quite a few models that we're now characterizing the growth trajectory and the metastatic pattern of spread. And finally, I'll just talk about patient-derived organoid models. Martin mentioned this too. Many people are very interested in these models, especially because of their potential for high throughput analyses like drug screens. And this is where you develop organoids or tumor organoids within matrigel domes inside of a dish. Here's an example of one model growing labeled with a fluorescent dye. And this is the same model treated with a drug. This is just data showing you a dose response to that particular drug as an example of the model. And we've been looking at optimizing these beautiful pictures or just new optimizations of different staining so we can track our favorite markers in these organoid models. And finally, I'll just mention that we're trying to establish an initiative, what we call PPOP, or a preclinical patient-derived organoid platform, which we proposed as a new and strategic initiative within the Faculty of Health and Medical Sciences, because we recognize now that we and many others are trying to work on organoid platforms, and we propose to bring all that expertise together to supercharge our translational research. So I'll say thanks for listening. I know it's short time. We created a video of our paper here, so I encourage you to go and visit it. And of course, I'll acknowledge many people around the world that contribute to our work. Thank you. I'll answer questions.
Thanks so much, Theresa.
Yeah.
Look, we are running relatively short for time, but I would invite people to ask their questions through the Q&A function. I might just kick off with a couple of really brief questions, if I could, Theresa. Mm-hmm. And they really relate to the sort of systems that you've developed as part of the Dame Roma Mitchell Cancer Research Laboratories, in particular, the sort of the PDEs that you've developed. I was just curious in terms of how reflected they are in terms of cellular composition with the primary tumor. And given that we now have exquisite technologies to look at single cells and single cell transcriptomics, can you give us a feeling of the relative sort of whether they are a high fidelity representation of the primary tumor?
They are, especially within that's why we developed it, so that you sustain the tissue architecture and the cellular complexity. Mind you, that you're looking at within fragments that you capture. So just like a biopsy from a patient, that fragment's going to have, different content. So we always have several replicates. It's really important to have replicates. But what we find is that we have, using the gelatin platform, it really sustains the viability of multiple cell types. The fibroblasts survive. You can see immune cells in there, tumor cells, and the architecture of that fragment does not break down, and that's the beauty of it.
That's fascinating. So in terms of just looking at the orthotopic models of breast cancer versus the one that you described, which is this mind xenograph model, profound ignorant question from my part, but in terms of actually the introductal, it's sort of almost putting those cells into that very defined space, what's the sort of rationale that sits behind that versus the sort of classical orthotopic models?
Well, it's because, breast cancers first develop within the ducts. They don't develop in the mammary fat pad. So it better recapitulates the natural environment where a tumor would arise. And also it's been shown that especially for estrogen receptor positive tumors, it sustains what we call the luminal phenotype of those tumors. That when you put an estrogen receptor positive tumor into the mammary fat pad, the microenvironment of the fat pad actually stimulates a more basal phenotype. So it changes the phenotype of that tumor, and putting it in the ducts better sustains what we call a luminal phenotype. Right. And also it's forcing, in the mammary fat pad, it just forms a big ball of cells. And they don't metastasize very well from that ball of cells unless you get invasion of blood vessels. Whereas when you inject them into the ducts, it first forms an in situ lesion. And then depending on the model, it'll start to invade the stroma. And then you see metastasis, which is what you would see in a patient. So it's just a better way to model disease progression than creating a ball of cells under the skin, basically.
And I presumably that when you actually challenge those tumors with chemotherapeutic agents, I presume you get quite differential responses from the sort of orthotopic bolus of cells versus the introductal.
You would think so, but it hasn't been formally tested yet.
Okay.
One of the things that we are doing some direct comparisons of that, but they do tend to metastasize better from the ducts, and so it's been really hailed especially as a better model for metastasis per se.
Wonderful. Look, thank you so much for your presentation. It's been fascinating, and it's wonderful to see the incredible work that you and the team are doing at the Dame Roma Mitchell Cancer Research Laboratories. Look, that brings us to an end of the session this afternoon. I'd like to thank both of our speakers, Martin Oehler and Theresa Hickey, for their excellent presentations. And before we close off, I'd like to remind people that these mini reviews are recorded and are available at the Adelaide Biomed City website. So if you go to the www.adelaidebiomed.com and under the banner webinars, you can see this seminar as well as all the past seminars from contributors across the precinct. I would also encourage you of those online to remind your colleagues that these webcasts are on Tuesday afternoons at 4:30, and just to build awareness of them. So please join us next week at the same time and same bat channel. Cheerio.